In this session, I will introduce something called views or BS genomes. Let's start off by loading in a genome package and get a DNA string. We have discussed this genome and this DNA string in the matching session, in the session on matching functionality from biostrings. It is the yeast genome, and we will run a match pattern on the yeast genome, or the yeast genome chromosome 1. Now, what we get out of this match pattern here, and we saw that in the earlier session, is something called a views object. Now, it's a views. We can see it refers to a specific DNA string object, and it has a start and an end. It looks a little bit like a DNA string set, but it also looks like an iRanges. And it actually turns out that underneath the hood, a views object is the same, or is represented as an iRanges. And we can get the ranges out of it by writing ranges, which is a single iRanges. We can check that these particular coordinate actually gets us the right nucleotides. So let's see here, 57932 to 57939, and that gives us the sequence we were matching for. So that fits. We can, even though it is a view and it's represented as underneath the hood as an iRanges, we can do stuff like run functions on it as if it was a DNA string set. In other words, we can do something like alphabet frequency on the view, and we get back the output of alphabet frequency on the DNA string set. Now, views are very powerful because they are a very efficient and fast way of representing what is basically sub-sequences or sub-parts of a bigger object. All we need to store is coordinates. We don't need to store the actual sequence. So a view consists of a set of coordinates plus an object they link into. That means we can do things such as we can shift the view by 10 bases. Now we get another sequence, which is the hit, but shifted 10 bases. And this computation was not really possible if all we had was the 8 bases of the hit. Let's see this in practice. We can do something like, we can get back at G-ranges by using the vmatch pattern function on this thing here, on the entire genome, VCA, and we get back a G-ranges, and we can instantiate a view object with the views function. We say we want the view object on the VCA genome, and the views are given as this G-ranges here. Now we actually get the DNA string associated with these coordinates. That's very powerful, and it allows us to easily represent many subsequences, such as promoters or exons. So let's take that into overdrive and compute the GC content of promoters in the yeast genome. So in order to get the promoters, let's load up annotation hub, which we should be familiar with by now. Let's do a query on our annotation hub for this specific version of the genome, and let's say refseq, or let's just say genes. Oh, I have to create my annotation hub first. It's downloading the annotation hub. Let's query it. and we get back two objects, SGD genes and ensemble genes. SGD is the Stanford or Servier Genome Database. It's the official yeast database, and then the other thing is from ensemble. Let's just take the first one. Let's use double brackets for downloading the genes. And now we want to get the promoters. Well, there's a nice convenience function for getting promoters in Bioconductor, which is just called promoters. Remember, promoters, and you can see that by examining the argument, gives you a 2.2 kp interval, 2000 downstream, 200 bases upstream. No, 2000 bases upstream and 200 bases downstream of the transcription start site. Now, we got a lot of like errors out here or warnings or whatever it is. Let's see here. Warning messages. So why is that? Well, it turns out that there are some of the yeast genomes that are right at the boundary of the genome. Let's look at element number 2 here that started around base 500 on chromosome 1, and it goes to base minus 1665. G-ranges complain when it gets like genome indices that are less than 0. And we get rid of this by taking the promoters and we trim them off, and basically we cut off anything outside the sequence length of the genome. So now we can see we have some promoters that are less than 2.2 kb long. Now, this was the promoters. Now we are going to instantiate a view of these promoters. So that's very simple. We say views, say VCA, and these promoters. Look how fast it was. And now we have the coordinates, but we also have the nucleotides in the different promoters. Now what remains is to get something like the GC content. We now basically run letter frequency on the views. We want C and G characters, and we want s.prop equals true. This was the GC content of all promoters in the yeast genome computed in like no time. Let's plot it. Let's do a density plot of these values. Okay, here we have a density plot. We have GC content on the x-axis, and you may remember from an earlier session that the GC content of the yeast genome is roughly 38%. Or was it 39? I recomputed here, but let's just say 38. And we can see the promoters don't really seem to have dramatically different GC content than the rest of the genome. That may be surprising, but remember that the yeast genome is a small genome. It's filled up with coding sequences and regulatory regions, unlike the human genome. So this was a small example of how powerful views can be. We will see in later session that we can instantiate views on other big objects. And this is a very, very efficient way of doing some computations.